Hello, in this video I'm going to talk about the life cycle of a somatic cell, and I'm going to talk a little bit about cancer too. So first of all, I want to talk about what the term cell life cycle means. So it is kind of what it sounds like it means. It talks about how a cell comes about and what the cell does during its lifetime, and it could possibly involve cell replication and cell death. So most of a cell's life is spent in a non-dividing state, right? In which case the cell is just being its little cell self. It's doing its job, whatever it is. If it's a cardiac muscle cell, it's beating away. If it's a neuron, it's sending action potentials. If it's a liver cell, it's detoxifying. Some cells divide, though. And why would we need cells to divide, right? So take a minute to think about this. Why would we need cells to divide? So maybe you came up with growth, right? It's one of the ways that we're able to grow physically larger, right? That's why babies are smaller than adults, is because adults have more cells, right? Or maybe you came up with repair, right? We need to replace dead and dying cells. So the cell division helps with that too. So this image just kind of gives you an idea of what the cell's life cycle is like, right? The green arrow is indicating the cell being its little cell self, doing its job, whatever it might be. And then up top in the sort of peachy area, it says dividing. So if the cell does divide, this would be the division state for the cell. So we're going to concentrate on somatic cells here, but first let's talk about what's the difference between a somatic cell and a germ cell. Right? So in the human body, we can take all of the cells in the human body and roughly separate them into two categories: somatic cells and germ cells. Somatic cells are pretty much all the cells in your body that are not going to become eggs and sperm, whereas the germ cells are eggs and sperm. They have different ways to divide. Somatic cells divide through mitosis, and germ cells divide through meiosis. We are going to consider what happens to a somatic cell in this video. Before a somatic cell can divide, it has to replicate its DNA. Another way of saying this is that before a somatic cell can divide, it has to replicate its chromosomes. So don't forget, chromosomes are just long pieces of DNA all packaged up, right? In either case, before the cell divides, we have to replicate the DNA. And this is important because if you don't replicate the DNA, then every time the cell divides, it will have half as much DNA, and this will lead to non-functioning cells. So if a typical somatic cell contains 46 pieces of DNA before the DNA is replicated, how many pieces of DNA are there after the DNA is replicated? If you came up with 92, you are correct. So after the DNA is replicated, the somatic cell will actually go into the division phase of the life cycle, which is called mitosis. During mitosis, what we're going to do is we're going to divvy up those replicated chromosomes. Okay. Mitosis is technically the process by which somatic cells divide up chromosomes. Okay. During mitosis, those 92 pieces of DNA are separated into 46 equal halves. So we're going to line up all of the chromosomes in the middle of the cell, okay? And then 46 pieces of DNA will be sent to one side of the cell, and the other 46 pieces of DNA will be sent to the other side of the cell, and then we cut the cell in half. And now we end up with two cells, each with 46 pieces of DNA, each with 46 chromosomes. And so now we have replicated the cell. Okay. The process of the cell actually splitting in half is called cytokinesis, right? This is the division of the cytoplasm. And what we get at the end of mitosis is two daughter cells. I don't know why they're called daughter cells, but they are. We get these two daughter cells, and these daughter cells are clones, right? They are clones of one another. They are clones of the starting cell. So before mitosis begins, we start off with one cell. And once mitosis and cytokinesis is complete, we end up with two identical cells. So how often do cells do this mitosis thing? Right? Well, it kind of depends upon what kind of cell you are and what stage of development the human body is in. You know, and in embryos, there's lots of mitosis going on in lots of different cells. You know, but in the human adult, only certain cells are going to undergo mitosis. For example, muscle cells. really don't undergo mitosis. Neurons don't undergo mitosis. So what this means is that as an adult, you have all of the muscle cells and all of the neurons that you will ever have. And so if they die, they're gone, and you can't easily replace them. You might be able to replace them using stem cells. So stem cells are capable of producing new cells, but we don't really have stem cells to make new neurons, and our stem cells for making muscle cells are really pretty terrible. So I wouldn't count on using those. So make sure you keep your muscles and your neurons healthy. Exposed cells are very good at doing mitosis. So for example, the surface of the skin is exposed, and the cells there are very good at undergoing mitosis. And we have stem cells there that are very good at replacing the cells in the skin. And the digestive tract is exposed. So the whole lining of your digestive tract is really good at doing mitosis to replace all of the cells there that are damaged and die, need to be replaced. You know, the average stomach cell lining the inside of your stomach only lasts about a day. And so we need to do lots of mitosis in order to get new cells to replace those dead and dying ones there. So whenever we talk about cell division, it's always important to talk about cancer. But before we do that, let's talk about cell division in tumors. Excessive cell division may lead to tumors. Another name for a tumor is a neoplasm. And a tumor is just an enlarged mass of cells that have abnormal cell growth and division. So cells are just continuously dividing without any type of regulation. Tumor does not mean cancer. There are benign tumors and malignant tumors. Benign tumors are not cancerous. They're not harmful. Malignant tumors are the cancerous tumors. These are harmful. Malignant tumors can be invasive, and what that means is that the tumor is actually invading into another tissue or organ, causing damage. An example of this is like a brain tumor. Brain tumors often form on the membranes that are on the outside of the brain called the meninges. And these tumors, as they grow, will press onto the brain tissue, causing it to malfunction. So that's an invasive tumor. Some tumors are malignant because they metastasize. What that means is that some tumors shed cells, and these cells travel through body fluids and then get stuck in another place, and then they start growing new tumors in that new location. So an example of this would be lung cancer. Lung cancer is highly metastatic, right? So the lung cancer begins in the lung tissue, but the cells are shed and they travel through like the bloodstream and the lymphatic vessels, and they can start growing new tumors really anywhere in the body. So malignant tumors are cancerous. What makes cancer cancer is to have uncontrolled cell growth that's having a deleterious or bad effect on the body. Okay. Can someone have cancer without having a tumor? Right? Have you heard of any cancers that don't involve tumors? Right? Maybe a lot of you are saying leukemia, which would be correct. So leukemia is a type of cancer that does not actually have a tumor. We still have uncontrolled cell growth having a bad effect on the body, but the cells aren't together in a clump as a tumor. So this is just a picture showing the process of metastasis, right? First of all, over here on this side of the screen, we start off with our single abnormal cell. And this abnormal cell is experiencing uncontrolled cell growth. We end up with a tumor, and then the tumor sheds cells into the bloodstream, and we start growing secondary tumors somewhere else in the body. That's metastasis. So what exactly causes cancer, right? Cancer is caused by damage to genes. A lot of people don't realize that, so you can star this and underline it and highlight it, but cancer is caused by damage to our actual DNA, right? We call these mutations. So do you inherit these genes already damaged from your parents, or do these genes become damaged later in your life, or both? If you said both, you're correct. So both can happen, right? Sometimes we do inherit damaged genes. Sometimes we acquire damage to our genes. So both can lead to cancer, right? So would you say cancer is caused by the environment or genetics, or can it be both? And again, the answer is both. So the environment does contain molecules called carcinogens, and carcinogens can damage your genes leading to cancer. But like I said, you can inherit damaged genes already. So both are possibilities. So acquired cancers are the ones that come about due to carcinogens causing the cancer, whereas inherited cancers are the ones that are caused when you inherit the genes already from your parents. A lot of cancers usually come about by both those things. So normally what happens is that cell division is very carefully regulated. We don't get cancer. So what I want to look at now is how these damaged genes can lead to unrestrained, unrestricted cell growth. Right? So normally cell division is very carefully regulated by lots of different proteins inside of the cell. Right? So proteins do most of the work for a cell, right? And we have lots of different proteins inside of our cells that are just really checking to make sure we don't get cancer, ultimately. And what they're doing is they are regulating cell division. They make sure that cell division only happens under perfect conditions. Okay? Now don't forget, proteins are made based on the information found in genes. Right? Anyway, so cancer is caused by mutations in genes whose proteins help to control the cell cycle. Right? So the way I usually think of it is like this. We have these little police officer proteins that run around in our cell and make sure that cell division is happening properly. Okay? And these police officer proteins do things like they check the integrity of the DNA. What does that mean? Well, they check the DNA to make sure that it's not damaged. Okay? And if these police officer proteins detect damaged DNA, they can do one of two things. Number one, they can start a process to fix the damage, right? But sometimes the damage is so bad, it can't be fixed. So then another choice that the police officer protein has is to initiate a process called apoptosis, which is programmed cell death. So sometimes it's better to kill the whole entire cell so that the greater good of the organism is maintained, right? Because if we have a cell that has damaged DNA and we let it replicate, we're going to get cancer. So these police officer proteins will initiate apoptosis, killing the cell, preventing it from unrestrained, unrestricted cell growth, and hence preventing cancer. The problem comes into play when the genes for these police officer proteins are damaged. An example of one of these proteins is called the p53 protein. And in almost all types of cancers, the gene for the p53 protein is mutated. So p53 protein is a tumor suppressor protein. What that means is that this protein normally stops tumors from forming. And like I said, it does things like checks the integrity of the DNA, can initiate repair processes if the DNA is damaged, or it can cause apoptosis to occur if the damage is too bad. But what if the gene for p53 is mutated? Well, then you're either not making p53 protein, or if you are making the p53 protein, it doesn't work at all. So now you don't have any police officers checking the integrity of the DNA, and now we have damaged DNA, right, maybe from a carcinogen, and there's nothing there to repair, to start the repair process. To fix the DNA, right? There's nothing there to initiate apoptosis if the DNA damage is severe. And so we have a genetically modified cell that's now going to just replicate, right? And then we get daughter cells, and then those replicate, and you get this unrestrained, unrestricted cell growth, cells multiplying, and you end up with tumors. So cell division goes on continuously, unchecked. Now, there are many genes that can be involved with cancer other than p53, right? But again, what's happening here with cancer is that the genes for proteins that regulate the cell cycle, those genes have become mutated. And now we've lost the ability to regulate the cell cycle, and cells replicate in an unrestricted, unrestrained manner.